Hello and welcome to Human Anatomy Prep. I'm Ty Gardner and I'll be your instructor. In this lecture we'll be taking a look at bone. Here's our basic mind map. We'll start by looking at bone as a tissue to understand bone structure. Then we can look at ossification, the formation of bone. Then we will look at different types of bones, with a focus on long bones like the femur. Finally, having an idea of bone shapes, we can look at landmarks of bones, the features that determine how they connect to each other, connect to muscles, and allow the passage of nerves and blood vessels. There are two forms of bone tissue: cortical or compact bone, and cancellous or spongy bone. Cortical is derived from cortex, meaning bark. Like the bark of a tree, a cortex is the outside layer of something. The alternative term, compact bone, refers to the dense, hard nature of this bone as compared to spongy bone. Spongy bone, named for the open spaces between the bone tissue, is located on the inside of bones. The technical term cancellous refers to the crossbars, trabeculae, that provide support while allowing spaces. Cortical bone is built on a tube-shaped unit called an osteon. The osteon is composed of a central, or Haversian, canal, and surrounding layers called concentric lamellae. Lamella means thin layer. Lamellae is the plural form meaning layers. Concentric lamellae are built around each other, all surrounding a center, the central canal. Here we see that if you stack tube-shaped osteons, you end up with spaces that aren't filled in. Interstitial lamellae, meaning between place layers, fill in the gaps between the osteons. Circumferential lamellae, meaning layers on the outside of a circle, smooth out the outside of the bone. So, what kinds of cells do we find in bone, and how do they relate to its formation and maintenance? There are 4 bone cell types, though it is also true to say that blood cells are formed in bones. Osteogenic cells are stem cells and can divide if more cells are needed. They become osteoblasts. Osteoblasts secrete new bone matrix and become trapped in a space within the matrix called a lacuna. Osteocytes, osteo equals bone, cyte equals cell, are the mature bone cells, trapped in a lacuna, that maintain bone. Osteoclasts, derived from white blood cells, dissolve and consume old bone to release calcium, or to repair bones. We've learned that osteoblasts make new bone matrix, but how are bones formed? Ossification is our term for bone formation, and there are 2 ways it happens. Intramembranous ossification, as its name suggests, occurs between 2 membranes. It occurs primarily in skull bones and leaves a sandwich with compact bone bread and spongy bone filling. The 2 membranes begin as embryonic connective tissue, but end up as what we call periosteum. Periosteum, meaning around bone, is a fibrous envelope surrounding each bone, regardless of ossification mode. Endochondral, endo equals in, chondro equals cartilage, ossification occurs inside a hyaline cartilage model of a fetus. Cartilage is converted to bone, and spongy bone is converted to compact bone to create the bone shape. All of the cartilage is converted except for the articular cartilage on the joint surfaces and the epiphyseal plate, an area where the bone can grow longer as a juvenile becomes an adult. Now that we have a general idea of where bone comes from, let's talk about the different types of bones. Short bones are, well, short. They tend to be cube-shaped like the cuboid bone of the foot. Short bones include the carpals in our wrist and the tarsals in our foot. Long bones are longer than they are wide and generally tube-shaped. The femur of the thigh is a good example. There are 22 long bones in one arm alone, including the humerus, radius, ulna, metacarpals, and phalanges. The long bones have a general structure that is of interest to us. We'll cover the three basic parts. The epiphyses are the ends of the long bone, the proximal epiphysis and the distal epiphysis. The proximal epiphyses of long bones are a place where we find red bone marrow. Flat bones are another. The diaphysis is the long, tubular middle portion of the bone. Inside the diaphysis, we find the hollow medullary cavity. Medulla equals middle, filled with yellow bone marrow, fat. Flat bones have a lot of flat surface. The sternum or breastbone is an example. Flat bones like the scapula and ilium provide attachment sites for many important muscles. What do you call a bone if it isn't short, long, or flat? Irregular bones like this vertebra have weird shapes. Many irregular bones, like the zygomatic bone and the maxilla, are located in the skull. Sesamoid bones are different. They form within tendons. The patella, or kneecap, is the biggest. Sesamoid bones can form in other joints, but the patella is the only one that all people have. To talk about the relationship between bones and muscles, nerves, or vessels, we need to know landmarks. There are a lot of skeletal landmarks you will talk about in lab. These basic terms will help. There is more than one term for a hole in a bone, but foramen, as in mental foramen, is the most common. A meatus, or canal, like the external auditory meatus, is also a hole, but is more like a tunnel through the bone. A fissure, like the superior orbital fissure, looks like a hole, but is actually a gap between two pieces of bone. There are many bumps. Tubercles, like the greater and lesser tubercles of the humerus, are usually rounded. The greater and lesser trochanters of the femur are like tubercles, but larger. A tuberosity, like the ischial tuberosity, tends to be lower and rougher than a tubercle. Our bones also need smooth surfaces at joints. Condyle is the most common term for rounded, smooth areas. We even have occipital condyles on the bottom of our skull that articulate with our top vertebra, the atlas. We also have usually flat, smooth surfaces called facets, such as the superior articular facets of our vertebrae. I hope this has been helpful. Your next stop is the video on the skeleton.